Hello, warm welcome to lecture 7th of cell biology, TYBSc cell biology paper 6th. Before that we have studied first two chapters, then this is chapter 7th of nucleus. We already studied some part of the nucleus. Only small part of this chapter is remaining, that is nucleocytoplasmic interactions. This nucleus and cytoplasm shows the transportation and the interaction between this through the nucleopore complexes. We are going to see in detail in this part. Myself, Dr. Sunil Patil from Loknethe Venkatragiri College, Panchavati, Nashik. Then let's move to the nucleocytoplasmic ratio. In this part, we are going to see the nuclear transports mainly. In the nuclear transport, we are going to see these points. First one is the nuclear transport receptors. The substances to be transported in and out of the nucleus, means between the exchange between cytoplasm and the nucleoplasm, the substances required some receptors. That receptor we are going to see in this part. This transportation, it is two-way. One is the nuclear import, means substances from cytoplasm into the nucleus, called as the nuclear import. Substances from nucleoplasm to the cytoplasm, that is nuclear export. Then protein shuffling, it is the additional part. This part we are going to see in this lecture. The first one is the nuclear transport. Nuclear transport refers to the mechanism by which molecules move across the nuclear membrane of cell. The entry and exit of large molecules from cell nucleus is tightly controlled by NPC, nuclear pore complexes. We have already studied the structure of nuclear pore complexes in the previous lecture. Although small molecule can enter the nucleus without regulation, but macromolecules such as RNA and proteins require association with the transport factors that known as the nuclear transport receptors. That factors called as the nuclear transport receptor, or they are also called the, it is a kind of nuclear transport receptor, karyopherines. They are either importins and exportins. From cytoplasm to nucleoplasm, this receptor called the importins, and from nucleoplasm to cytoplasm, these are called the exportins. So in this figure, nuclear import and nuclear export is shown. In the import, the receptors are required. We are going to see in detail the nuclear export and nuclear import. Before that, some important things. Protein that must be imported to the nucleus from cytoplasm carry nuclear localization signals. It is called the NLS, that are bounded by importance. Means the protein that to be imported from cytoplasm to the nucleoplasm. It is called the NLS, nuclear localization signals. Mainly they are called the importance. And nuclear localization signal is a sequence of amino acid that is acts as a tag. To the tag, substances are attached. They are diverse in the composition and most commonly hydrophilic. The proteins, tRNA, transfer RNA, and assembled ribosomal subunits, they are exported from nucleus due to association with exportin, which bind to signal sequencing. That signal sequence is called the sequences of amino acid. They are called the nuclear export signal, NES. It is NLS for the import, and for the export it is called the nuclear export signals and nuclear localization signals for the import. For the export, nuclear export signals. The ability of both importin and exportin to transport their cargo is regulated by the small RAN. Guanosine triphosphatase are enzymes, GTPase. They are enzymes that bind to molecule called as the guanosine triphosphate, which they then hydrolyze to create guanosine diphosphate, means GTP converted to GDP, and this thing releases energy. It is very common. And RAN is a different conformational depending on the whether it is bounded to GTP or GDP. Means RAN bound to GTP, it is different thing, and GDP, it is different thing. In its GTP bound state, RAN is capable of binding the karyopherines. When it binds to GTP, having the stored energy, it is called as the karyopherine. The importin releases cargo upon binding to RAN GTP, while exportin must bind RAN GTP to form ternary complex with their export cargo. So it is the procedure. The dominant nucleotide binding state of RAN depends on whether it is located in the nucleus, that is RAN GTP, or the cytoplasm, RAN GDP. In cytoplasm it is RAN GDP and it is RAN GTP in the nucleoplasm. It is the difference. In this way it carries the substances. So this is RAN cycle, this is nuclear import, very simple thing, and nuclear export thing. We are going to see stepwise nuclear import. First, importing proteins bind their cargo in the cytoplasm, after which they are able to interact with nuclear pore complex and pass through its channel. Once inside the nucleus, means in the nucleoplasm, interaction with RAN GTP causes a conformational change in the importin. Means there is change in the importin able to bind with the cargo, means substance to be carried, and causes it dissociate from its cargo. The resulting complex of importin and RAN GTP then translocates to the cytoplasm, translocate in the cytoplasm, where a protein called RAN binding protein, RAN BP, separate RAN GTP from importin inside the cytoplasm. Separation allows access of GTPase activating protein that binds RAN GTP and induces a hydrolysis of GTP to GDP. So we have already seen this thing. The RAN GDP produces from the process now binds to nuclear transport factor, which return to the nucleoplasm. Now in the nucleus, RAN GDP interact with the guanine nucleotide exchange factor GEP, and it becomes GTP, resulting again separation of GTP, resulting again RAN GTP formation, the beginning of the new cycle. So it is in this way nuclear import takes place. I think you got it. Then another thing is nuclear opposite to this only. Nuclear export roughly reverse of the import process. In the nucleus, inside the nucleoplasm, exportin bind the cargo and RAN GTP and diffuses through the pore to the cytoplasm, where the complexes are dissociated. So it is reverse of that. RAN GTP binds GAP, hydrolyzes GTP, and resulting RAN GDP complexes is restored to the nucleus, where it exchanges its bound region for GTP. Hence, whereas the importin depend on the RAN GTP to dissociate from their cargo, exportin require RAN GTP in order to bind to the cargo. A specialized mRNA export protein moves mature mRNA to the cytoplasm after post-translational modification is completed. The translocation process is actively depend on the RAN protein, although the specific mechanism is not yet understood. Some particularly common transcription located in the cytoplasm, located near nuclear pore, they facilitate the transportation. So this is the import cycle, this is export cycle. In the import cycle, this is the RAN GDP. It causes conformational changes in the importin. Then this is changed importin according to substance. That substance bind to the importin. Then this importin moves through the nuclear pore complex. These are the annuli of the nuclear pore complex. There are also conformational changes, and this complex comes into it. Then due to RAN GDP, there is again conformational changes which releases the importin. Then RAN GDP binds with the importin. That importin again ready for the next cycle. Reverse of this happen in the export cycle. Means this is exportin, RAN GDP causes changes in the exportin and the substance bind to this. So RAN GDP separates, and RAN GDP with this as it is GTP, and it goes through the nuclear pore complex in the cytoplasm. Then it releases energy, it separates the RAN, it becomes GDP now, GTP becomes GDP, RAN GDP, and this is the exportin, the export substance, and exportin separates. This exportin again back to the nucleoplasm for the next cycle. In this way, import cycle and export cycle takes place. So export of tRNA is also dependent on the various modification it undergoes, thus preventing export of improperly functioning tRNA. This qualified control mechanism is important due to the tRNA central role of translation, where it is involved in adding amino acid to growing peptide chain. The tRNA exporter in vertebrate is called as the exportin T. Exportin T binds directly to the tRNA cargo in the nucleus and process promoted by the presence of RAN GTP. Same thing, only it binds directly to the tRNA cargo. Mutation that affect the tRNA structure inhibits its ability to bind to exportin T and consequentially to be exported, providing the cell with another qualified control step. So this thing happen in the exportin. So protein sapling, it is, although it is not in syllabus, but it is part of this. Many proteins are known to have both NES and NLS. You know this. And localized signal and exporting signal, and thus shuttle constantly between nucleus and cytoplasm or cytosol. In certain cases, one of these steps, nuclear import and nuclear export, is regulated often by post-translation modification. Protein shuttling can be assessed using a heterokaryon fusing assay. So I think you got this part. Next lecture it is on the cytoskeleton, lecture 8. So see you again. Thank you very much.